press the bell icon on the YouTube app and never miss another update.
Namaste children. So let us continue with this topic, neural control and coordination. In our previous class we discussed generation and conduction of nerve impulse. Just we'll glance once again. So in the generation and conduction of nerve impulse, ಇದು 5 marks ಗೆ ಬರುತ್ತೆ. So when neurons are at rest, when neurons are at rest, what happens? We see outside positive charges, inside negative charges. So this is polarized state. So polarized state means outside positive, inner negative charge. Axolemma ಹೊರಗಡೆ ಪಾಸಿಟಿವ್ ಚಾರ್ಜ್ ಇರುತ್ತೆ ಎಕ್ಸ್ಟ್ರಾ ಸೆಲ್ಯುಲರ್ ಫ್ಲೂಯಿಡ್ ಅಲ್ಲಿ, ಎಕ್ಸೋಪ್ಲಾಸಮ್ ಅಲ್ಲಿ ನೆಗೆಟಿವ್ ಚಾರ್ಜ್ ಇರುತ್ತೆ. ಬಿಕಾಸ್ ಏನಕ್ಕೆ ಅಂದರೆ ಅಟ್ ರೆಸ್ಟ್ ವಿ ಸಿ ಸೋಡಿಯಂ ಅಂಡ್ ಪೊಟ್ಯಾಷಿಯಂ ಪಂಪ್ ವಿಲ್ ಬಿ ಆಕ್ಟಿವ್. That means when neurons are rest, 3 sodium ions will move out, with in turn we get 2 potassium ions inside. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, 3 sodium ions will efflux and 2 potassium ions will influx. And axolemma or neurilemma is permeable to potassium ions and neurilemma is impermeable to sodium ions and negatively charged ions. So due to this, and also you can write one more, voltage gated channels of sodium and potassium is closed. So ಈ ಎಲ್ಲ ಕಂಡೀಷನ್ ಇಂದ ಏನಾಗುತ್ತೆ, ಡ್ಯೂರಿಂಗ್ ರೆಸ್ಟ್ ದ neurilemma outside it contain positive charge, inner it contain negative charge. ಇಷ್ಟು ನೀವು ಒಂದು ಪಾಯಿಂಟಲ್ಲಿ ಬರೀರಿ. ನೆಕ್ಸ್ಟ್ ಇನ್ನೊಂದು ಡಯಾಗ್ರಾಮ್ ಹಾಕಿ. After explaining this, or with a one diagram only you can explain. I will tell you once again. So when stimulus is applied, the potential which is developed between the membrane at rest is called resting potential. It is minus 70 millivolts. So when you apply stimulus, this region will excitatory region. So ಎಲ್ಲಿ ಸ್ಟಿಮ್ಯುಲಸ್ ಅಪ್ಲೈ ಮಾಡ್ತಿರೋ, ಸೊ ಅಲ್ಲೇನಾಗುತ್ತೆ, ವಿ ಸಿ ಇನ್ಸೈಡ್ ಪಾಸಿಟಿವ್ ಚಾರ್ಜ್, ಔಟ್ಸೈಡ್ negative charge. Inside positive charge, outside negative charge. ಏನಕ್ಕೆ ಅಂದರೆ when the neurons are stimulated, we see opening of voltage gated channels of sodium. So ಅವಾಗ ಏನಾಗುತ್ತೆ, ಎಕ್ಸ್ಟ್ರಾ ಸೆಲ್ಯುಲರ್ ಫ್ಲೂಯಿಡ್ ಇಂದ both side we see the entry of sodium ions that reverses the polarity. So inside becomes positive, outside becomes negative. but adjacent area B will be in a polarized state only. B will be in a polarized state. ಹಾಗಾಗಿ ಇನ್ನರ್ ವಿ ಸಿ ನೆಗೆಟಿವ್ ಚಾರ್ಜ್, ಔಟರ್ ವಿ ಸಿ ಪಾಸಿಟಿವ್ ಚಾರ್ಜ್. ಯಾವಾಗ ಸ್ಟಿಮ್ಯುಲಸ್ ಅಪ್ಲೈ ಆಗುತ್ತೆ, ಎಕ್ಸೈಟೇಟರಿ ರೀಜನ್ ಅಲ್ಲಿ ವಿ ಸಿ ರಿವರ್ಸಲ್ ಆಫ್ ಪೊಲಾರಿಟಿ. ಸೊ ದೆನ್ ಮಿನಿ current circuit or electric circuit develops from A to B and B to A. Once it completes, the stimulus will shift to B region and A will become repolarized state. So this happens, and that potential difference which develops in the region at stimulus is called action potential. It's called action potential. After the circuit completes, it will shift to B region. So due to that, so again agutte, once it completes, it will be B region, it will be A region, so then once the circuit completes, the stimulus will shift to B region and this region will become repolarized due to the opening of voltage gated channels of potassium and closing of voltage gated channels of sodium is going to be closed. It would be closed. Immediately once stimulus shifts to next region, previous region alli enagutte, sodium gated channels will be closed and potassium will be open, so that efflux of potassium takes place, again restoring back the negative charges inside, positive charges outside. So this is how the stimulus, once it generates here or once action potential is generated, it will shift from one region to next region till it reaches the axon terminals, till it reaches the axon terminals. So ನೀವು ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಒಂದೇ ಇಲ್ಲ ಮೂರು ಡಯಾಗ್ರಾಮ್ ಹಾಕ್ಬಿಟ್ಟು you can explain. So in your exam you can draw this one diagram. So illen ಮಾಡ್ತೀರಾ, when neurons are at rest, ಈ ಒಂದು ಡಯಾಗ್ರಾಮ್ ಹಾಕಿ you should write, when neurons are at rest we see negative charge inside, positive charges outside, because membrane neural lemma is permeable to potassium ions and impermeable to sodium and negatively charged molecules, and sodium potassium active pump will be working, and so voltage gated channels for sodium and potassium will be closed. ಆ ಥರ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಬೇಕು. ವೆನ್ ನ್ಯೂರಾನ್ಸ್ ಆರ್ ರೆಸ್ಟ್ ಅಂತ ಹೇಳಿ ಒಂದು ಡಯಾಗ್ರಾಮ್ ಹಾಕಿ, and write one more diagram where stimulus is applied, and this is excitatory region. So then change the diagram, outside negative. So when stimulus is applied, that region will become excited and membrane will become permeable to sodium ions. That permeability of sodium ions reverses the polarity in the excited region, so that inner becomes positive, outer becomes negative, and adjacent to that will be in a polarized state. Hagagi mini electrical circuit will develop from A to B, A side to B side and B side to A side. And that potential difference which we developed due to this reversal of polarity at depolarization region is called action potential. ಅದು ಬರಿಬೇಕು. So when neurons are stimulated in the excited region, you see reversal of polarity due to the membrane will become permeable to sodium ions. Voltage gated channels for sodium is going to be opened. and that region is depolarized and mini circuit will develop with the adjacent region which is in this region will be in depolarized state and this region will be in the polarized state. Mini circuit develops from depolarized to polarized and from A to B here and B to A here. Once the circuit is complete, the stimulus will shift to B region. Stimulus will be shift to B region like that, and once stimulus shifts to B region, the previous region will become repolarized due to the opening of potassium gated channels and closing of sodium gated channels. This tara you explain madbeku, or you can write the diagram what they have given in your textbook and explain step by step. Understood? That is how once the action potential is developed, then till it reaches the nerve ending, it is going to be conducted along the length of the axon. In this class, we will discuss ಇಲ್ಲಿ ತನಕ ನಾವೇನು ಓದಿದ್ವಿ till it reaches axon terminals ಓದಿದ್ವಿ. In this class, we will discuss from the axon terminal how it transfers to next neurons under synapses. So, synapses or synapse, it is a junction between two, it is a junction between two functional neurons that is synaptic node or terminal end of axon, synaptic node or terminal end of axon with the dendron of other neurons. Nothing but it is a gap. This is synaptic node and this one is dendron. This is dendron. This is synaptic node. This one is axon. So what is synapse? It is a junction between two functional neurons, that is between synaptic knob or terminal end of axon with the dendron of other neurons. This junction is called synapse. This junction is called synapse. Sherrington or he described synapse is a contiguity. What is contiguity? It is nothing but connection without continuation. Sherrington ಏನು ಹೇಳ್ತಾರೆ, synapse is a contiguity. That means there is a connection between two neurons without any continuation. ನೋಡಿ ಇಲ್ಲಿ ಗ್ಯಾಪ್ ಇದೆ, continuation ಇಲ್ಲ, ಇಲ್ಲಿ ಗ್ಯಾಪ್ ಇದೆ, ಆದರೂ ಕೂಡ we see connections with a chemical substance called neurotransmitters. So Sherrington told synapse is a contiguity, that means connection without continuation. Andre anatomical connection is absent whereas physiological connection is present. What is anatomical connection? So iದ್ರ ಮಧ್ಯೆ ಏನಾದರೂ ಈ ತರ connectivity ಇತ್ತಂದ್ರೆ ಇದಕ್ಕೆ ನಾವೇ that is anatomical connection. Whereas in synapse anatomical connections will be absent. Only physiological, ಅಂದ್ರೆ movement of ions takes place. movement of chemical substance takes place. ಹಾಗಾಗಿ there we see physiological connection, not the anatomical connections. So that is about synapse. It is a junction between two functional neurons, between the synaptic knob of one neuron with the dendron of another neuron. Next we will go with structure of synapse. We know that usually it will be inner negative charge, outer positive charge. This is axon. This one is synaptic node. Mitochondria. Receptors. This is So this is structure of structure of synapse. This is axon terminal which is ending with synaptic node, and action potential will be carried. This is action potential. We see a calcium channels also. So this is axon. It terminates with synaptic knobs. That knobs contain secretory vesicles. Inside the secretory vesicles we see neurotransmitters, and we see mitochondria which provides ATP molecules for neurotransmitters, and the membrane of the synaptic knob is called presynaptic membrane. And next we see dendron. The membrane of the dendron is called postsynaptic membrane. The receptors for neurotransmitters are present on the membrane of postsynaptic. It is present on the postsynaptic membrane. That is, they are present on the membrane of dendrons. So, in competitive case, receptors are seen in postsynaptic membrane or presynaptic membrane, or in both. And the receptors for the neurotransmitters are seen only on the membrane of postsynaptic dendron. That is, and the junction between the presynaptic membrane and postsynaptic membrane is called synaptic cleft. The junction between presynaptic membrane and postsynaptic membrane is called synaptic cleft. So, where the neurotransmitters are released, This is about structure of synapse. So, it is having presynaptic membrane of synaptic knobs and postsynaptic membrane of dendrites. Inside the synaptic knobs we see secretory vesicles, inside that we see neurotransmitters. Now we will discuss how the impulse is going to transmit from axon terminals to dendron. When action potential reaches, when nerve impulse reaches the synaptic knobs, we see the opening of voltage gated channels for calcium. Calcium start moving into the synaptic knobs. As this calcium moves into the synaptic knobs, the secretory vesicles will move towards the membrane, and we see the exocytosis of secretory secretory vesicles will come and adhere to the axolemma, and exocytosis, they release neurotransmitters to the synaptic cleft. See, once action potential reaches the synaptic knob, we see the opening of calcium voltage gated channels. So, calcium inside move adaga, they moves secretory vesicles to the axolemma, where exocytosis of that secretory vesicles takes place, releasing neurotransmitters to the synaptic cleft. So, ಇಲ್ಲಿ synaptic cleft ಅಲ್ಲಿ release ಆಗಿರ್ತಕ್ಕಂಥ neurotransmitters ಏನಾಗುತ್ತೆ, they will come and adhere to the receptors present on the dendron. The neurotransmitters will come and adhere to the receptors present on the dendron, and action potential will be regenerated in the dendron. Next, from the dendron till the axon terminal, ಮತ್ತೆ ನಾವು previous class ಅಲ್ಲಿ discuss ಮಾಡಿದ್ವಿ, ಅದೇ ರೀತಿ it will be conducted. So, this is how it functions. So, synapse ಏನಾಗುತ್ತೆ, when action potential reaches the synaptic knob, we see the opening of calcium voltage gated channels. This calcium pushes the secretory vesicles towards the axolemma, where we see exocytosis of that secretory vesicles seen, which releases neurotransmitters to the synaptic cleft. These neurotransmitters will adhere to the receptors present on the dendron. When that neurotransmitters added to the receptors present on the dendron, we see opening of sodium gated channels. Opening of sodium gated channels. When sodium enters, again depolarization occurs and action potential will be generated. So, like this impulse will transport from axon to dendron. And some of the neurotransmitters, example we have acetylcholine, it is a neurotransmitter. Acetylcholine is a neurotransmitter. Some of the neurotransmitters again go back to the secretory vesicles. Once the impulse is transported to the dendron, we see enzyme choline esterase that splits acetylcholine acted by this enzyme and splits into acetate plus choline. See, once the impulse is transported from axon to dendron, the enzyme choline esterase will be released that act on acetylcholine and make it to convert into acetate and choline. Again that acetate and choline will reabsorb into the vesicles, and again they are reused This is how the function takes place between the axon and dendron. So, when action potential reaches the synaptic node, secretory vesicles will adhere to the postsynaptic membrane or neurolemma of the axon, and exocytosis takes place, releasing neurotransmitters to the synaptic cleft, and neurotransmitters will adhere to the receptors present on the dendron and results in opening of sodium gated channels, so that action potential will be regenerated in the dendron. Like this, impulse will be carried from axon to dendron. Understood? So, this is how the function we see at the synapses. So, let us discuss the types of synapses. So, synapses are divided into two types, anatomical and physiological. Nimage physiological is very important, but idu tilkondiri ashte. Anatomical we have three types. Axo-axonic, axosomatic and axodendritic. Axo-axonic andre nimge gottagate. So this is axon. This is axon, and you can see like this. This is one more axon. Axonic means there is a synapse between axon of one neuron with axon of another neuron, which is less common, that is axonic. Axosomatic antandre, so this is again this is axon. This is soma. If the axon is near to cytosol or cell body, then it is called axosomatic. It is called this is cell body and this is axon. Axosomatic is a type of synapse between the cell body of one neuron and axon of another neuron. Between axon and axon, it is between axon and cell body, this is. And axodendritic, which is very common, axodendritic is very common. So we see this is axon. It will end with a this is dendrites. This is axon. So, axodendritic are more common synapses where we see a gap between axon of one neuron and dendrite of another neuron. This is about anatomical classification of synapses. Then we will go with physiological classification. So, under physiological classification, we have two types: chemical synapses and electrical synapses. We have two types, chemical and electrical synapses. So, separately I will take. There are two types of synapses, that is chemical and another one is electrical. So, the chemical synapses, roughly I will draw the diagram. This is synaptic node. This is dendrite, axon. So, in the chemical synapses, we see presence of neurotransmitters. like acetylcholine. The impulse from the axon will be transferred to the dendrite of next neuron through a chemical substance called neurotransmitters. Neurotransmitters are seen in the synaptic cleft, whereas in electrical synapse, neurotransmitters are absent. Neurotransmitters are absent. So this is axon. From the axon, the impulse will be shifted to dendron without any help of without any help of neurotransmitters. So illenagate impulse will shift from axon to dendron without any neurotransmitters. So that is seen in electrical synapse. Adike nodi synaptic knob alli yavdu nanu secretory vesicles kuda bardilla. Secretory vesicles are absent because in electrical synapse there is no role of neurotransmitters. The impulse will be conducted from axon to dendron by a induction method, or it jumps from axon to dendron. There is no role of neurotransmitters. Hakagi neurotransmitters are absent. And synaptic cleft will be we have a the junction between axon and dendrite is called synaptic cleft. It is of 10 to 20 gap between axon and dendron will be 10 to 20 nanometer. And the gap ಜಾಸ್ತಿ ಇದೆ. ಇಲ್ಲಿ electrical synapses ಅಲ್ಲಿ it is very less. Gap between synaptic cleft is just 0.2 nanometer. ನೋಡಿ just 0.2 nanometer irodrinda impulse will directly shift from axon to dendron. And we can write the next point, delay in the synaptic cleft or delay in the conduction of impulse. Here we see very or no delay in the conduction of impulse. Illi yake delay agutte? Delay in conduction of impulse or transfer of impulse. Why here we see delay andre once the action potential reaches the axon terminal, the secretory vesicles or neurotransmitters has to release to the synaptic cleft, then they are added to the receptors present on the dendrite. Hagagi it will take time, whereas in electrical synapse, as there is absence of neurotransmitters, there is no role of neurotransmitters and impulse will directly enter from axon to dendron. Hagagi there is no delay in transfer of impulse. There is no delay in transfer of impulse. And the other difference is the faster conduction of impulse is seen in electrical and little bit lesser in chemical synapses. And chemical synapses are unidirectional. Always the impulse will be transferred from axon to dendron. Always from axon to dendron. It is unidirectional. Whereas in electrical, it is bidirectional. Bidirectional anta antashna nimage gottagute. It is that impulse can be transferred from axon to dendron or dendron to axon or dendron to dendron or axon to axon. So in electrical impulse we see bidirectional. but in chemical it is always unidirectional. And in vertebrates, in higher vertebrates, we see chemical synapses only. In very few area we see electrical synapses in our body. Majority of the synapses present in our body is chemical synapses. This type of electrical synapses is seen in lower invertebrates. It is seen in invertebrates and in some lower vertebrates. Understood? That's the difference between chemical and electrical synapses. New main at tilkoladu. See, in chemical synapses we see a role of neurotransmitters, but neurotransmitters are absent in electrical synapses. The gap between the presynaptic and postsynaptic membrane is called synaptic cleft. That will be 10 to 20 nanometer in the chemical synapses, and it is 0.2 nanometer in electrical synapses. There is delay in the transfer of impulse from axon to dendron. Synaptic delay is seen in chemical synapses, that is not seen in electrical synapses. Here we see faster rate of conduction. In chemical synapses we see delay in the conduction, whereas in electrical synapses we see faster in the conduction of impulse or in the transfer of impulse from axon to dendron. And chemical synapses they are always unidirectional, electrical are bidirectional. These synapses are seen in vertebrates and these are seen in invertebrates. So that is the difference between chemical synapses and electrical synapses. Then coming to neurotransmitters, they have not given this in your textbook, but just tilkondirri ನೀವು ಅಷ್ಟೇ. So what are neurotransmitters? They are biochemical substance helps to transfer impulse from one neuron to another neuron. What are neurotransmitters? They are biochemical substances helps to transfer impulse from one neuron to another. We have two types of neurotransmitter. One is excitatory neurotransmitters, another one is inhibitory neurotransmitters. Excitatory example we have acetylcholine or norepinephrine, they all example for excitatory. Inhibitory we have serotonin and gamma amino butyric acid. They are not given in your textbook, but you tilkondirri, because sometime in your need they may ask like acetylcholine is a excitatory or GABA is a inhibitory, ee tara kelbodu. So excitatory alla adu ಏನು ಮಾಡುತ್ತೆ ಅಂದ್ರೆ, see, this is synaptic node and this is dendron. So when acetylcholine is released into the synaptic cleft, they allow the opening of sodium voltage gated channels. ಯಾವಾಗ acetylcholine ರಿಲೀಸ್ ಆಗುತ್ತೆ ಸಿನಾಪ್ಟಿಕ್ ಕ್ಲೆಫ್ಟ್ ಅಲ್ಲಿ, in postsynaptic membrane. In postsynaptic membrane, we see the opening of sodium gated channels that depolarizes the membrane in the dendron, and action potential is going to be generated. That is how it acts as a excitatory. It acts as a excitatory. Excitatory means it helps to transfer impulse from one neuron to another neuron. So inhibitory, so when sodium influx takes place, the reversal of polarity agutte, depolarize occurs, and action potential is generated. Inhibitory enagutte, that is synaptic node. So when GABA is released, that is gamma aminobutyric acid is released, we see opening of chlorine gated channels which is negative charge. We see opening of chlorine voltage gated channels. Chlorine is negative charge. When chlorine enters into the dendron, there is no reversal of polarity, there is no conduction of impulse. So that's why they act as a inhibitory neurotransmitters. See, excitatory neurotransmitters ಏನು ಮಾಡುತ್ತೆ, when they adhere to the postsynaptic membrane, they open the sodium gated channels. Sodium are positive in ion, they are positive ions. Hagagi in postsynaptic membrane we see depolarization. That means there is generation of action potential. In that way it carry the impulse from one neuron to another. Inhibitory enagutte, this gamma aminobutyric acid, they allow the opening of chlorine voltage gated channels. Chlorine are negative charge. So at rest you know that inside negative charge, outside positive charge irutte when neurons are at rest. So here, because as the chlorine are negative ions, there is no depolarization, there is no conduction of impulse. Like this they inhibit the transfer of impulse from one neuron to another neuron. This is about excitatory and inhibitory types of neurotransmitters. So in this class, for your exams, what you have to remember means, you should remember synapse is a junction between two functional neurons, that is between axon terminal or between synaptic knob of one neuron with the dendron of another neuron. Next, there are two types of synapses, chemical synapses and electrical synapses. So generally now structure kuda odidu chemical synapses. The chemical synapses alli we have presynaptic membrane seen in the axon and postsynaptic membrane seen in the dendron. So between these two we see a gap called synaptic cleft where neurotransmitters are released. So this, in chemical synapses neurotransmitters are present, in electrical synapses neurotransmitters are absent, and there is delay in the transfer of impulse in chemical synapses and there is no delay in electrical synapses. Chemical synapses are always unidirectional and electrical synapses will be bidirectional. So ಈ ಎರಡು ಡಿಫರೆನ್ಸ್ ನ ನೀವು ಡಿಫರೆನ್ಸ್ ನ ತಿಳ್ಕೊಂಡಿರಬೇಕು between chemical and electrical synapses, and structure of synapse kuda nimige 3 marks ge barbodu diagram. So that completes the generation and conduction of impulse from dendron till the axon terminal and from axon terminal to the next neuron. The next class will discuss about the properties of neuron. Okay, thank you.